Hello everyone. Today our topic of discussion is sterilization technique. Plant tissue culture media rich in sucrose and other organic nutrients support the growth of many microorganisms like bacteria and fungi. प्लांट टिश्यू कल्चर में जो सुक्रोज एंड ऑर्गेनिक न्यूट्रिएंट्स होते हैं वो माइक्रो ऑर्गेनिजम्स की ग्रोथ में हेल्पफुल होते हैं. On reaching the media, these microbes generally grow much faster than the cultured tissue and finally kill it. ये जो माइक्रोब्स होते हैं कल्चर मीडिया पे बहुत जल्दी ग्रो करते हैं और हमारे कल्चर टिश्यू को डिस्ट्रॉय कर देते हैं. द कॉन्टेमिनेंट्स में आल्सो गिव आउट मेटाबॉलिक वेस्ट विच आर टॉक्सिक टू प्लांट. जो मेटाबॉलिक वेस्ट इनकी प्रोसेस से निकलता है वो आल्सो टॉक्सिक होता है प्लांट टिश्यू के लिए. देर आर टू जनरल precautions are there. First is not to share plant tissue culture working area with the microbiologist and pathologist. Second is remove contaminated cultures from the culture area as soon as detected. The possible source of contaminations are culture vessels, nutrient media itself, the explant environment of transfer area, instrument used to handle plant material during inoculation and subculture, the environment of culture room and the operator. Types of sterilization. Basically there are three types of sterilization is used. First is dry heat, second wet heat, and third is filter sterilization. The method of sterilization is different for living matter than tools and glassware. Now we discuss about dry heat. Dry heat method is used for glassware, tools and other articles which do not get affected by high temperature. Media and plastic ware cannot be sterilized by this method. Now we talk about method how which dry heat is used. Glassware tools are put in a container or wrapped in paper or thick aluminum foil and then they are placed in a dry oven and sterilized for a period of not less than 3 hours at a temperature of 140 to 160 degrees Celsius. Next is wet heat. It is the most popular method of sterilization for both equipments and media. Equipments and media is autoclaving at 121 degrees Celsius with a pressure of 15 psi pound per square inch for 15 minutes. Modern autoclaves are capable of providing saturated steam treatment ranging from 70 to 132 degrees Celsius, which is a pressure of up to 25 psi. All the vessels containing medium should be placed vertically and should not be filled more than 40% of their total capacity. There is a relation between amount of medium to be sterilized and duration of autoclaving, or we can say that there is a direct relation, directly proportional the amount of medium to the duration of autoclaving. The table is showing volume, container and the medium sterilization duration. Filter sterilization. Only liquids can be sterilized by this method. This method is not used for plant material and other things. It is also called cold sterilization because solution sterilized without heating or autoclaving. It is used when a solution or medium cannot be sterilized by autoclaving. It is the property of a filter have porosity 0.22 to 0.5 micrometer to retain all the microorganisms and make the solution free from microbes. This is commonly used where thermolabile or heat sensitive chemicals like plant growth regulators, GA, IA, abscisic acid and some biochemicals, enzymes for protoplast isolation, antibiotics or nutrient solution for specific experiment are used to avoid decomposition during autoclaving. So in these filter sterilization is used. There are two type of filters are commercially available like glass and membrane filter like nitrocellulose membrane. Glass filters like sintered glass filters and borosilicate are available in different grades from G1 to G5. G5 have 1 to 2 micrometer porous size is used for bacteriological purposes. The grades are classified by maximum pore size, which is obtained by measuring the pressure at which the first air bubble breaks away from filter under certain conditions. These filters have a porous glass disc for filtration of liquid and gases as filter media, which is non-corrosive and reusable. The filters are resistant to heat, solvent and detergent. Membrane type filters. Membrane type filters have different diameters and porosity from 0.22 to 0.45 microamp and are available as disposable and reusable filters. Membrane filters can be connected through tubing or syringe to pass solution or gases. The picture is showing glass filters and membrane filters. The first picture is showing setup having membrane filter. In the picture there is a funnel, clamp, membrane filter and there is a reservoir to collect the filtrate. The second picture is showing glass filter. There is glass funnel, sintered glass disc and a suction flask to collect the filtrate. How filter sterilization is done is discussed in following steps. Filters are wrapped in aluminium foil or paper and then autoclave at 121 degrees Celsius for 15 minutes and then are taken to laminar airflow bench. The solution is placed in funnel having glass type filtered or syringe having membrane type filter. The solution is allowed to pass through filters under suction pressure. A membrane fitted or glass tube filled with cotton may be placed between vacuum flask and suction pump to avoid entry of air, and the sterilized filtrate is collected in the flask. Sterilization of tools. How tools are sterilized. Surgical blades and scalpels are not sterilized by dry heat because the high temperatures make the cutting edge dull. Articles including spatula and forceps immerse in 80% volume by volume ethyl alcohol until required. Sterilized during use by frequent immersion in alcohol and flaming. Bead sterilization is also available for sterilizing tools. Bead sterilization working on the principle of dry heat. Use of fumigation like formaldehyde and sulfur is avoided during sterilization. It is used sometimes to clean the labs or working area from microbes, but the place not used for 3 to 4 days following fumigation. Culture rooms fumigated after the removal of all the cultures. Generally, the whipping of working place with the 80% alcohol is sufficient for regular use. Sterilization of explant. All the explants sterilized before transferring onto the medium. A suitable size explant sterilized by any one of the following solutions. First is 1 to 4% solution of sodium hypochlorite, that is commercial bleach. Second, 7% saturated solution of calcium hypochlorite. Third is 1% solution of nitrate solution. 4 is 1% solution of bromine water. Fifth is 70% ethyl alcohol solution. Sixth is 0.1 to 0.2% mercury chloride. Seventh is 10% hydrogen peroxide solution. The types and concentration and duration of exposure of the particular sterilant to be used are depend on plant material that is used for the experiment. The picture is showing sterilization of explant. The explant in laminar airflow washed with 70% of ethanol for 5 minutes, then rinsed 3 times with sterilized distilled water. Again then washed with sodium hypochlorite at 5%, 10%, 20% for 5, 10, 20, 30 minutes depend upon percentage. Then again rinsed with 3 times with sterilized distilled water, and then the outer layer of shoot is cut and transferred into a MS media. In many cases, a drop of liquid detergent or wetting agent like T-Pol, Tween 80 is incorporated into the sterilizing solution to enhance the penetration and effectiveness of sterilizing agent. Material should be thoroughly washed several times with sterilized distilled water to ensure proper removal of sterolide. All the sterilized material and plant parts should not be touched directly by hand. Sterilized forceps should be used for this. Last, hands and benches are made aseptic by ethanol 80% before starting the work. Thank you.